Hello and welcome to my YouTube channel. Today I'm going to discuss about bacterial cell and its cell wall structure. In my previous video I have talked about the gram staining procedure. As a result, we have discussed about the gram positive bacteria which appeared as purple while the gram negative bacteria appeared as pink. This color coding of the gram positive and gram negative bacteria is depend on the structure called cell wall, and therefore we can say the cell wall is a essential uh, component of a bacteria which we need to remember. So let's start. First of all, uh, just I'm going through with the size of a bacterial cell. So there are great variation in size of a bacteria. Each bacterium measures about 1.25 micrometer to 2 micrometer in diameter. The smallest bacteria is Mycoplasma genitalium, while the giant bacteria is sulfur bacterium called Teichoic volutens. It is largest among all the bacteria. Now, we have certain uh, shapes of both gram positive as well as gram negative bacteria. Likewise, and if we see a bacteria in oval to round shape, we call them cocci. If we see a bacteria with uh, a rod shaped structure, we call them bacilli. So cocci may be arranged in pairs, cocci may be arranged in chains, cocci is in cluster, tetrad, or sarcina. And same with bacilli. Bacilli can be arranged in singly or in chains or comma shaped or in Chinese letter pattern. So these are basically, we have two major shape of bacteria, that is cocci and second is bacilli. If a bacteria is of oval to round shape, we call them cocci, and bacteria with a rod shape structure, we call them bacilli. Now, here is you can see the structure of a bacterial cell. Here, the outermost layer is called capsule. Some of the bacteria is having capsule; they are known as capsulated bacteria, and those we the bacteria which do not have this outermost structure called capsule, they are called non-capsulated bacteria. The bacteria which are having this structure is called capsulated bacteria, and the bacteria which don't have this structure called non-capsulated bacteria. Just beneath the capsule we have cell wall, and the innermost layer of a bacterial cell is called cell membrane. uh, Inside the cell membrane we have certain organelles like mesosomes, nucleoid, ribosomes, and some inclusion bodies. We have their cytoplasm. One more structure is flagellum. Flagellum is a thread-like structure which help to bacteria in the movement. So the bacteria which is having this flagellum, they are motile bacteria, and the bacteria which do not have this flagellum, they are non-motile bacteria. So here we need to remember is the outermost is capsule, then cell wall, then cell membrane. Inside the cell membrane we have cytoplasm, right? Next, now the main focus of this lecture is on cell wall. So cell wall is a rigid solid covering which provide the shape and structural support to the bacterial cell. Cell wall lies between plasma membrane and glycocalyx. Glycocalyx is a outer part. Periplasmic space occurs between plasma membrane and cell wall. Cell wall protect the bacterial cell against bursting in hypotonic solution. In gram-positive bacteria, the cell wall is about 20 to 80 nanometer thick, it is single layered, and it is smooth. In gram-negative bacteria, the wall is 88 to 12 nanometer thick, complex, and two-layered. The outer layer is also called outer membrane. It consists of lipopolysaccharide, lipid, and some proteins. The single layered cell wall of gram positive bacteria and inner wall layer of gram negative bacteria is made up of peptidoglycan, protein, and non cellulosic carbohydrates, lipids, and amino acids. Bacteria can be divided into two major groups called gram positive bacteria and gram negative bacteria based on the gram staining. The gram positive and gram negative bacteria differ in the appearance of cell wall. The cell wall of gram negative bacteria is multi-layered structure and is very complex, while gram positive bacteria contain primarily single type of molecule and is much thicker than gram negative bacteria. Now, the essential component of cell wall is peptidoglycan. In the bacterial cell wall, there is a one rigid layer that is primarily responsible for strength of the wall, that is peptidoglycan. In gram-negative bacteria, additional layer is present outside of this layer. The rigid layer of both gram-negative bacteria and gram-positive bacteria is very similar in chemical composition and is called peptidoglycan. This layer is thin sheet composed of two sugar derivatives named as N-acetyl glucosamine and N-acetylmuramic acid. In short, they are NAM and NAG, NAM and NAG. These constituents are connected to form a repeating structure, glycan tetrapeptide. Basic structure of peptidoglycan is a thin sheet in which the glycan chains formed by sugars are connected by peptide crosslinks formed by amino acid. Glycosidic bonds connecting the sugar in the glycan chains are very strong, but these chains alone cannot provide rigidity in all directions. This is important. Peptidoglycan is present only in bacteria. N-acetylmuramic acid is never found in the cell walls of archaea and eukaryotes. In peptidoglycan, the glycan proportion is uniform with only the sugar NAM and NAG being present. And these sugars are always connected in beta 1 to 4 linkage. More than 100 different peptidoglycans types are known, and the greater variation among them occurs in interbridge. Peptidoglycan constitutes 7 to 80% wall in gram-positive bacteria, while the lipid content is very little. In gram-positive bacteria, 10 to 20% of wall formed of peptidoglycan. But in gram-negative bacteria, the higher lipid content, that is 20 to 30%, is present. One more essential component is teichoic acid. Gram-positive bacteria have acidic polysaccharide called teichoic acid attached to their cell wall. But in gram-negative bacteria, teichoic acid is not present. The term teichoic acid includes all wall, membrane, or capsular polymers containing glycerophosphate and ribitol phosphate residues. Teichoic acids form receptor sites and surface antigen because they are negatively charged. Teichoic acid are partially responsible for the negative charge of cell surface and may function to effect passage of ions through the cell wall. Certain glycerol containing acid are bound to membrane lipid of gram positive bacteria because these teichoic acids are intimately associated with lipid. They are being called lipoteichoic acid, as they are associated with lipid. Therefore, the name is lipoteichoic acid. In gram-negative bacteria, outer membrane is covered by lipopolysaccharide. The lipopolysaccharide consists of complex polysaccharide covalently linked to lipid. Lipid portion of lipopolysaccharide refers to as lipid A, which is not a glycerol lipid, but instead the fatty acid are connected by esteramine linkage to a disaccharide composed of N-acetyl glucosamine phosphate. Fatty acid commonly found in lipid A is lauric, myristic, palmitic, and beta hydroxy myristic acid. Here you can see the difference between gram positive and gram negative bacterial cell wall. The thickness is gram positive bacteria is positive cell wall is thicker than gram negative bacteria. About the periplasmic space, it is absent in gram positive bacteria while it is present in gram negative bacteria. Lipids in gram positive bacteria, we can observe a small amount of, or maybe it may be absent in gram positive bacteria, while lipids are present in gram negative bacteria. Teichoic acid is present only in gram positive bacteria, however absent in gram negative bacteria. The thickness of peptidoglycan layer is 16 to 80 nanometer in gram positive bacteria, while it is 2 nanometer in gram negative bacteria. This is the difference between gram positive and gram negative bacterial cell wall. So for today, this was the lecture. In next video, I will be completing the principle of gram staining, how they gram positive and gram negative bacteria retain the dye and appears in different colors. Till then, thank you very much for listening. Thank you.